It's now my great pleasure to introduce our next speaker, Dr. Leonie Ringrose. So Dr. Ringrose started a scientific journey with a degree in molecular biology between Norwich and Marseille universities, after which she worked as a patent examiner for the European Patent Office in Munich. She then came back to academia and did her PhD in the lab of Dr. Francis Stewart at the EMBL in Heidelberg. She stayed at the EMBL for a short postdoc and then moved to the Institute of Human Genetics in Montpellier. However, probably Heidelberg stole a little piece of her heart because she came back after 2 years and for a senior postdoc position at the Center of Molecular Biology at the Heidelberg University. Next, she moved again and joined the IMBA in Vienna as a junior group leader, where she opened a lab to study the balancing of polycomb tricorax and their dynamic and recruitment technology. In 2015, she was awarded a full professorship for quantitative biology at the Humboldt University in Berlin. But before moving there, she did a sabbatical in the group of Martin Howard to get a bit more familiar with mathematical modeling of gene regulation, and that probably laid the conditions for a wonderful paper on bistable regulatory system behind polycomb and thorax interaction that was published last year. Finally, despite being an experienced PI, a professor, a very active contributing member of the community, she did not leave the bench completely, and she's still performing experiments, analysis, and technique development. And to put it in her own words, on lab days, I start my day with bench first, even before coffee. I get my experiment running, and everything else has to fit in the incubation times. Leonie, we're really happy to have you here and very much looking forward to your talk. Thank you for joining us, and the floor is yours.
Thank you very much. It's a great honor to be invited here, and it's still kind of blowing my mind that people are all over the world joining us here. Yeah, I'm going to talk about the topic of epigenetic regulatory elements and ask the question of whether they are all the same. And in the title of my talk, you have this concept of take, remember, and give back. And I'll get into what I mean by that. Now, I'm going to stay at a fairly conceptual level today, but if there are specific questions on any details, we can get into that during the questions. So let's start with this concept. What is a code? You probably can think of different sorts of codes. Perhaps one of them you'll think of is the genetic code. I think it's a real shame that non-coding nucleic acid is defined by what it doesn't do. It doesn't code. But one thing I'm very interested in is whether these, what we call non-coding nucleic acids, either DNA or RNA, do they have a code? And can we predict what they do by looking at their sequence? And let's start with something that Myron covered absolutely wonderfully, so I can go through this quickly. We all think of epigenetic regulation as something that stabilizes cell identities. And if it goes wrong, many things go wrong. But also, as Myron pointed out, and as many of us are becoming sort of getting into this idea, it also allows flexibility. So we have the stem cell, which is flexible anyway. We have stably determined cell types, but it could happen late in development or after development is finished that the cell gets a new signal, whatever that is. And some genes have to respond, even though they're kind of pinned down by epigenetic memory. They have to respond, others don't. So there's a change in cell status or something that cell does, but the cell doesn't change its identity. And something I've been very interested in and that I'm going to talk about today is how do some genes escape memory to allow flexible response to signals? And how do others know not to respond? And to get into that, we're going to talk about my favorite epigenetic regulatory elements. Myron also mentioned this, so it's great that, you know, we have a kind of mainly fly and a mouse talk, because I'm going to go from the fly to the mouse. So yeah, I love these elements. I sort of fell in love with them in 1997, I think, and still working on them. So they are epigenetic regulatory elements, PRE-TREs. And in mouse and fly, we have about 1,000 target genes, or more than 1,000 target genes, perhaps now. Staying with the fly, the way these elements work is sort of the classic idea is that the promoter will be bound by a transcription factor that will determine whether the gene is off or on. And depending whether it's off or on, either the polycomb or the trithorax group will get the upper hand. They will take that information from the promoter. The promoter in the fly example then is free of transcription factor. The transcription factor disappears. The element remembers what it was doing and gives back to the promoter. And by remembering, what I mean is the element has to survive with its state intact through DNA replication and mitosis, which are very different molecular challenges. So this is the kind of classic model, the dogma. And we know PRETREs in flies best for their role in maintaining this epigenetic memory. But there's another side to the story, which is that many of the target genes that we know from the fly show much more dynamic regulation. And by this I mean, first of all, that the transcription factor that regulates the gene doesn't disappear, it's still there. And that transcription factor, or several of them, may change their information that they give to the promoter continuously. And then we come to the question, what does the PRETRE then take from the promoter? What does it remember and what does it give back? And you can see that if the transcription factor is still there, this very same system has potential to be an amplifier or a modulator of gene expression rather than a pure memory element in the strictest sense. And that's an idea that I'm going to explore with you today and that I find very interesting. And I think that understanding the full repertoire of what this system can do is going to be essential for understanding genome-wide polycomb trithorax function in flies and in mammals. Now, I want to point out that even just in the fly, if we stick with the fly, the polycomb trithorax response elements that we know, and that we know they do that job, they have very different sequence compositions, by which I mean, for example, they have different orders and numbers of the motifs that we know are important for their function. And so, I would like to know whether these differences influence PRETRE function. And that is, do they influence actually quantitatively the strength of any of these three things that we know those elements do in the fly? And can we actually study that? And then if we can, is there a PRETRE code? Meaning that if we see a piece of DNA sequence and we know the developmental context, we know the promoter, can we then predict what that piece of sequence is going to do in that given situation? Okay, so we need to systematically dissect those properties. We want to know whether DNA sequence does actually affect those properties. And finally, I'm going to have a look at some mammalian PRETREs in that same kind of framework. Now, this is why I'm going to change gear, and some of you will like this, some of you will hate it. I think you'll all like this. This is okay. We're going to use reporters integrated at the same genomic locus so that we don't have effects of the local sequence on the behavior. And most of today's talk is going to be about mathematical modeling of the input from the promoter and the output. And let's see how that goes down. Maybe it's an unusual topic for this session for this audience, but let's see. Okay, here's the model. This work is published in Nature Communications last year. This is the work of Jeanette Reinig in the lab and an ex-PhD student from Vienna, Martin Howard, with whom I collaborated, and my own work. So this is a simple mathematical model that combines a polycomb trithorax response element and a promoter. The PRETRE part of it is based on published work from many, many people, which came up when physicists first started getting interested in this as a bistable system. It has three possible states, and the active and silent states are stable, whereas the intermediate neutral state is unstable. So it's a bistable system. There's nothing new about that model. The promoter is very simple. It's simply an array of potential binding sites, and the amount of output of the promoter corresponds to how many of those binding sites are bound. The new thing about this that hasn't been done before in the modeling world is to have these distinct units coupled in a way that is regulatable, and that turned out to be essential for modeling real data. So that's the model. What can we do with it? The user puts things in. We vary the promoter input to simulate developmental or experimental changes. And then we fit the parameters of this to see, can I actually see in my model what I've already seen in the experiment? Now, this is the simulation algorithm. Again, I won't go into detail, but I'd be happy to talk more about this. The important thing here is that we have real time. Why do we have real time? Because we're going to replicate the system in a way that reflects the real cell cycles that we know from Drosophila development. And when we replicate, we do two things. We remove all the transcription factors from the promoter, so we strip the promoter. That's to reflect the observed dissociation of most transcription factors as the cell goes through mitosis. And we remove modifications from 50% of the nucleosomes in the PRE-TRE. That's to reflect the partitioning randomly of modified nucleosomes to daughter strands. So it's a very, very simplified system. The important thing about it is that the timing of the replication cycles reflects the cell cycles that we know from Drosophila development, so we can start getting an idea of timescales. The promoter activation, that's what we do to the system. We can change that to reflect any gene that goes up or down in any given at any given stage of development. And the thing that we now want to fit are the PRE-TRE parameters. And that's the things that describe how well is it coupled and what does it do. And just to sort of bring that home to you, there are 4 free parameters. One is how well do these two things talk to each other? How long does it take for silent or activated states to stabilize? This A represents active, M represents silent. And they build up like an avalanche, if you like, because of feedbacks in the system. And that is something that can be very different from one situation to another. And the last one is the switchability of the system. And it's the arrows here, which enable the system to forget what it was doing and switch directly from one state to the other. And that turns out to be very important in fitting the model to data. So what are we doing? We're going to be dissecting PRE-TRE properties. I'm going to show you examples today of 2 different, very, very different types of PRE-TRE regulation in flies, one which gives stable memory and the other that gives dynamic regulation. And the question will be, can that same simple model recapitulate both of those different types of behavior? So I'm going to go back to a real classic experiment from the 1990s, which was what really gave PREs, the PRE-TREs, the name of memory elements. And that is, we take a reported gene without and with its normal PRE-TRE. This is what the gene normally looks like, and we just take out the PRE-TRE. What happens when we don't have the PRE-TRE element is that at about 5 hours of development, the repressors in the anterior part of the animal disappear and the reporter becomes activated. When we have the real construct, which is really existing in the genome, which includes the PRE-TRE element, the reporter, although the promoter is trying to get activated, the element gives a memory of the silent state from the previous developmental stage. And to cut the long story short, we set that model up with the promoter without the PRE-TRE, and then we fitted the parameters of the PRE-TRE so that we get that very similar memory. So we make the repressors disappear at this stage, and we then see that the PRE-TRE gives the memory of the silent state. As I said, I'll be very happy to answer questions about the details of this. I just want to give you this as an example. And the important take-home message from this is that both in the experiment and the model, the PRE-TRE gives a stable epigenetic memory of silencing. We can run this simulation up to 10 days, 20 days. This will remain stable. It's a very strong memory. Okay, what about dynamic regulation? I promised you I'd show you an example of that. And that's an example of a gene which gets switched on in the larva. That's after 4 days of development, much, much later, and it's very dynamically regulated. The gene has a PRE-TRE, which we've characterized previously very thoroughly. And the difference here is that this gene is continuously regulated by transcription factors and that it makes a very dynamic pattern in this structure of the animal, which is the eye disc, which is going to later on in the larva, it's formed, and in the adult, it makes the eye. Now, the pattern of that gene, when we make a reporter gene that includes the entire genomic DNA up to the second exon, we fuse that to GFP, we look at the GFP mRNA pattern, we get a kind of gradient across the eye disc, which you see here. And we can then quantify that into a little gradient pattern here, which is what we then use for fitting the model. So in the animal, the important thing is that we can do this with and without the PRE-TRE. So without the PRE-TRE, if we now look at the profile, there is a gradient, but it's rather flat. When we add the PRE-TRE to that, what happens is it fine-tunes that gradient, it sharpens it, it makes it higher where it's high and lower where it's low. So again, to cut a very long story short, we implemented that entire developmental cascade in the model so that we could then ask, can we get our model PRE-TRE to do the same? And the answer is yes. Here's the output of the model, where we see that in the model without the PRE-TRE, we have a rather flat gradient. And merely by implementing this very simple model and by changing these parameters to make the PRE-TRE do what we want it to do, we see that we can make this fine-tuning of the gradient. So again, the take-home message, what's important is that in both the experiment and the model, the PRE-TRE fine-tunes this dynamically generated gradient. So it's a completely different situation, but it's the same model. And just to summarize that, the simple model can recapitulate both. Of course, you'd like to know what is the difference. Well, we're doing a lot of different things here. We're changing 3 things at a time. We're giving it a different promoter, obviously. We're at a very different stage of developmental timing, but we also had to change the parameters of the PRE-TRE, and that's the thing I'm most interested in. So what's the difference here? In the stable situation with the embryo, we use one set of parameters to fit. In the dynamic situation, a different set. This is the parameter that's most different. That's our P5, and that's the one that governs the spontaneous switching of the element. So in the case where we get this stable memory, the element doesn't switch very often on its own. When we have the dynamic regulation, to fit that, we needed to have a very fast switching, meaning it doesn't remember very well what it's doing, meaning it can still listen to the promoter. And then it can give this gradient output. Now, does DNA sequence determine these PRE-TRE properties? I've shown you here that we change so many things that we can't really tell whether it's the DNA sequence that's really doing this, because there's so many things that are different. So we did a very simple and very old-fashioned experiment, which is the following. We have our reporter gene that I've shown you already. This is what this reporter gene does when it has its own PRE-TRE element, the one that belongs to it. If we then take the element that gives the stable silencing in the embryo assay, and we replace that with, we replace the correct one with the wrong one, if you like. What's going to happen? Are we going to get now the same behavior, or is something different going to happen? This was actually quite surprising. This is the experiment that you've seen already, with the correct polycomb trithorax response element. This is the model. Now, it took us about half a year to do this experiment, where we cloned the other PRE-TRE, we made the flies, we looked at the disc, and quite surprisingly, this gives a variegated pattern in the disc, but which still has a gradient. Now, to do the same experiment in the model took about a minute. You just have to change the parameters and run the simulation again, and quite surprisingly, without any fitting at all, but using the same parameters as for the memory assay, we get a very similar behavior out of the model as what we see in the experiment. And the important take-home message here is that in both the experiment and the model, the PRE-TRE converts the smooth gradient into a variegated gradient. By variegated, I mean you have lineages of cells which are off, lineages of cells that are on. It's the same as what you see here. And by gradient, I mean the onness in the place where the gene is more highly expressed is higher than the onness in the place where it's repressed. So this is a mixture of digital and analog output with kind of smooth input. So that's very interesting. So we are proposing that the inherent switching frequency, because the P5, the parameter that causes switching, is the one that allows this model to be fitted to the data, we propose that the switching frequency depends on PRE-TRE sequence and determines the properties. And what I'm showing you here is the real configuration of these motifs in each of those two elements. Now, we don't know why yet. One of them switches and one of them doesn't. We don't even know if it really does do that, but that's the hypothesis. And I think this brings us towards a PRE-TRE code. And it gives a kind of fresh perspective on that, because generally PRE-TRE code, people think about what recruits those proteins. But, you know, I think there might be something really interesting hiding in these sequences that makes them more or less stable in some way. I'll be very happy to talk about what I mean by that during the questions. So here's the overview so far. I've talked about how we can systematically dissect these properties with experiments and modeling. The sequence does indeed determine the properties in the case of this fly experiment. And so for the last few minutes, I'll talk about mammalian PRE-TREs and to ask whether they have different properties. And I don't mean different to the fly, I just mean different to each other. Do they also have personalities? So let's talk about this for the last few minutes. PRE-TRE sequence is not conserved between flies and mammals. All the things we know and love and that Myron introduced so very nicely are very well conserved between flies and mammals. But the sequences that those proteins bind to, we can find no similarity when we look for them in the mammal using the principles we know from the fly. So that's already interesting. And if you then look within mammalian PRE-TREs, these are published elements that have been shown to respond to polygon regulation, we also see that they look very diverse. And so the question is, do different mammalian PRE-TREs have different properties? And what we're going to do again is a reporter assay where we have a GFP this time hooked up to a minimal promoter called MP. We have different candidate mouse PRE-TREs. Importantly, we integrate them into the same genomic locus in mouse ES cells. This is work that was started by Johanna Trupke when she was doing her PhD with me. It's been continued by Natalia Chadala, who's done the master's thesis and is now doing the PhD. And there's input as well from Björn and Mark, who are our collaborators. So what you see now when you take these 23 different candidate PRE-TREs and you look at what is the reporter RNA level by looking at the barcode which is attached to each one of them, you see that the RNA expression ranges over orders of magnitude, which is already quite surprising. So that raises the question, are they just enhancers? It could be that we've just picked up enhancers. And we don't think so, because we don't see any correlation between the reporter level, which is on this axis, and the endogenous RNA level for each of those genes. Then we'd like to know, are they PRE-TREs, in fact? And we think they probably are, because they do recruit histone modifications consistent with their range of expression levels. And just while Myron was talking about CPG islands, I added a little bit of information here that is actually the most highly expressed ones that contain CPG islands, and the ones with no CPG islands are repressed, and that doesn't stop them from recruiting H327 methylation. We don't know what that means yet. I'm going to stick with these two red ones, which are two very, very well characterized polycon trithorax response elements that have either been published or we've characterized them. The question then, what do they actually do to the minimal promoter? And we were quite surprised when we looked on the single cell level, because we found that they cause variegation. If you look in the microscope, you see that the minimal promoter is rather uniformly repressed. The engrailed PRE added to that gives some cells which are more highly expressed, and the HOXD1112 gives a strong variation in expression. If you quantify this, you see that the minimal promoter is uniform. These are fluorescence values from single cells over 500 cells. The engrailed PRE-TRE starts to add some brighter spots, and the HOXD1112, it lights up on fire, but it doesn't all light up to the same extent. Now, I'm going to cut a very long story short with modeling this, but we can actually use this very same model to recapitulate that data. So we don't have to make any different assumptions about how it works, except for one single assumption. That is the following. We have to prevent the PRETRE from listening to the promoter. I'll be very happy to talk about what I think that means. Basically, the PRETRE ignores the promoter. It doesn't copy the promoter. It does its own thing. The rest of the model is the same. PRETRE is bistable. It gives back to the promoter. And when we then do that, we can recapitulate exactly the data from the model by only adding an activation bias to the HOXD1112 PRETRE, which I think comes from the CPG island. So I'm going to conclude then with giving you the take-home message here is that mammalian PRETREs indeed show different degrees of variegation and activation in this assay. And the two PRETREs are shown here with some of the motifs that we find in them. You can see they're very different. Again, we don't know why those motifs contribute to that difference, but we're in a position to address that. Now, I'd like to point out that variegation is something that an animal has very little use for. Why would I want a gene that switches randomly on and off? But if we think about what we learned from the model, an element that variegates in one situation, an artificial situation, has a very strong memory in a different situation. So I think that the variegation is telling us something about the memory properties of these elements. So that's something we'll be looking at in future. Again, towards a PRETRE code, we're definitely not there, but something that we now have the tools to address. So I'm going to summarize there. We've done experiments and models, particularly important that we put these into the same genomic locus for the experiment and that the model dissects these different properties. We did find in the fly, when we put a PRE into the two different PREs into the same situation, we do get different outputs, so they do have a mind of their own. Mammalian PRETREs definitely have different properties, and it seems to be very interesting in both cases that those properties are perhaps defined by their switching behavior. Can we define a PRETRE code? Definitely not yet. But I think we have the tools to address it. And what we need to do is not put completely different PRETREs into those assays, but ones with minimal changes, so we can start to pull out what depends on which sequence motif. And just at the end, I asked this question at the beginning: how do some genes escape memory? How do others know not to respond? And I think we have good evidence now that the sequence of those elements itself contributes to this. It's not the whole story, but it definitely contributes. And it contributes by influencing each of these properties perhaps differently. And what we'd like to understand in future is, how do I understand that PRETRE code? So I'm going to finish there and thank all the people who are brave enough to be part of my lab in Berlin. The people whose names you see are people whose work I've shown today. These people also contributed to the work that I've shown you. I'm going to thank these people for funding, and I thank you for your attention.
Thank you, Leonie, for the amazing talk. Really interesting. I loved it. Maybe I'll start. I'll take the first. I noticed that when you mentioned the model in the stable condition, it was mostly, if I'm right, in cycle 14-15 in flies, which is, let's say, relatively homogeneous regarding cell identities. I was wondering whether you have looked into the possibility of, modeling the changes in cell identity that can come up afterwards.
Yeah, it certainly would be technically doable. Yeah, that's a great question. We only focused on that switch in the UBX promoter that happens exactly at cycle 14, basically. So the first 13 cycles are the very fast ones. Cycle 14, everything slows down. UBX changes it comes on, and then the repressors disappear later. So yeah, I mean, the model is out there, right? The model is available on GitHub, and you can really address any question. You just have to know what question you want to address. Yeah, I'm certainly changes in cell identity that happen later, yeah, actually, we sort of did do that because the eye disc gets specified around about, in the larval instar, which is several days into development, and we had to get that to work in the model. We had to have the coupling switched off until that moment, because if you couple the thing too early and the gene is silent, the PRE will just silence everything, right? So yeah, we did actually address, the eye identity, the genes that switch on in the eye. Yeah, so it's yeah, it's
yeah, it makes sense.
Yeah.
Let's Anneke,
Yeah. I was wondering if you could help me understand better, like what could be behind this B5 parameter, like the different, how well it's remembered in different cases. I'm thinking, like, could it be different compositions of PRC complexes that behave differently? Could there be a demethylase on some sequences on others? Like, how are you thinking about that?
Question, yeah. I promise you that me and Hanneke did not actually communicate before this session, because I've actually got a slide for that, if I may show that. Can I?
Go ahead, please.
That's it. I was hoping you would ask that question. Let's go back. Yeah, just a different representation of the slide of the motifs. So this one switches fast. It's got this particular pattern of motifs. This one switches slower. It's got a different pattern of motifs. One of the things I think is really intriguing is this, and it's shown here. It's a long run of GA. And GA is bound by the GAGA factor. The GAGA factor is a nucleosome remodeler, and it binds cooperatively. The more sites you've got lined up next to each other, the more it's going to get in there and take out nucleosomes. So I think that might be part of it, that the GAGA factor can get in and it can kick off nucleosomes. It basically demethylates, deacetylates, de-everything, because it kicks nucleosomes out. So that's one thing we're super interested in. There's other aspects, that one might think there's more of one polycom than the other, but because of this kind of avalanche behavior in the system, which is something we looked into a couple of years ago in the 2019 paper, basically, once you start tipping it towards one direction, it's really hard to kind of delicately bias it. Yeah. So I think you've got to kick everything off. So my best bet is on the GAGA factor. And that's a testable hypothesis, right? Because we can put the element in different numbers of GAGA factor binding sites. We can do GAGA mutations. It gives us something to work with.
Measure turnover.
Yeah. Having said that, every single prediction I've ever made without using the model, I've ever made just by thinking I know how it works, in this project has always been wrong. So, modeling helps you think what to do, but it doesn't give the right answer. You have to do the experiment. The good thing about modeling is it's free. So, we can do this without spending any money and hopefully do a meaningful experiment. I stopped sharing. Yeah, I love that question. It's, what is it? I think that's testable now. We have to look at the right things. In the mammalian system, I think it's going to be essential to look at the single cell behavior, because that's very rich data that allows us to fit the model not just to an average but to a spread of high and low values, and that's a very, very simple experiment to do, but it gives really a lot of information, more than just averages. So yeah, I hope that answered your
thank you.
Great. I had a couple of questions coming in, and I see a pattern that is mostly, so I'm reading thank you, but it's echoed already in another question. Amazing work as usual. Thanks, Leonie. Does the distance between mammalian PRE and the core promoter matters in your model in terms of gene expression?
Yeah, wonderful question. Thank you, Peng. I see that there. Thank you so much. I hope so, yeah. I think, my answer is always going to be the same. I can do that in the model really, really easily. It will take me 2 minutes this evening to make a prediction with the model. It'll take us a year or so to build those constructs, but I think it would be worth doing. Absolutely. Yeah, it's a great experiment. Yeah, and then one would expect that the distance might affect that communication. It might change the how well they communicate with each other. If they communicate in cis across, being next to each other. If they communicate some other kind of fancy way, then maybe not. Yeah, that's a great question. Yeah, wonderful. Thank you.
I will go with the last one from Lauren Boulanger. Great talk. Do you think it could be possible to synthesize a TRA instead of making mutations in an existing one?
Thank you. Do you want to come to my lab and do that? Yeah, no, that's a fantastic question. Beautiful. Yeah, that's where we want to go, right? Yeah, we'd like to be able to design the thing and know, exactly what it would do. Giacomo Cavalli did this with Drosophila years and years ago, just before it became possible to do genome-wide stuff. He was doing some absolutely beautiful experiments with Jérôme Desjardins in, I think he was already in Montpellier at that time. Is that right? Or no, he wasn't. Yes, he was. I can't remember. No, but they published a beautiful paper where they made a synthetic PRE in Drosophila. And, that sort of stands as a shining example. And people kind of stopped doing that stuff when genome-wide things became possible. It's obvious why, because it's much easier to generate a genome-wide data set than to go and fiddle around making a reporter gene, that might not work, right? But I think in the end, we've got to get back to, synthetic reporters, design principles, design stuff, and test it, and see what comes out in a very systematic way. Beautiful question, yeah.
Yeah, the final proof that you understand something is when you can build it from scratch.
Exactly, you build it from scratch. Engineering principles. Yeah. Wonderful.
So, thank you very much, Leonie. It was really, really great, and thank to everyone that attended.